Hello everyone, my name is Darien Lozon and I'm in my last semester as a master's student in Wildlife and Fisheries Resources here within the Davis College Division of Forestry and Natural Resources. One of the chapters in my thesis investigated the effects of land use practices on freshwater turtle bioaccumulation. In this presentation, I'm going to be discussing some results of that study regarding mercury levels found in snapping turtles and how these detected levels raise concern for daily harvest limits in the state of West Virginia. The tricky thing about mercury, though, is that it occurs naturally in the Earth's crust, which is fine until it becomes bioavailable when soil is disturbed by practices such as tilling, digging, and so on. This disturbance then exposes the mercury to bacteria and plankton that then convert it to methylmercury, which is the most toxic form of mercury. Because snapping turtles have long life expectancy and practically eat anything they can fit into their mouths, they are more susceptible to mercury bioaccumulation, and most of the mercury found in their bodies is methylated. We as people take in mercury from the food we eat as well. However, there are stricter guidelines on what concentrations are safe for us. The U.S. Environmental Protection Agency has a set daily consumption limit of 0.0001 milligrams per kilogram. That is 1/10,000th of a milligram. For wildlife, on the other hand, methylmercury limits were established in the Clean Water Act, but only for fish. This is likely because people are more likely to consume fish. We need to keep in mind, however, that snapping turtles are one of the more common turtle species harvested for the commercial meat trade internationally. To bring it into a more local perspective, the current daily harvest limit for snapping turtles in West Virginia is 10 individuals per day, with no more than 20 individuals kept at a given time. Keeping these health and harvest regulations in mind, we ask the questions, what are the mercury levels in snapping turtles within a typical mixed-use watershed in West Virginia? And more importantly, are they within the safe human consumption limits? To address these questions, we collected 16 blood samples and 30 nail samples from 33 individual snapping turtles in the Upper Deckers Creek watershed, located in Preston County. The Upper Deckers Creek watershed covers 7,778 hectares and is a predominantly forested habitat that has been fragmented by agriculture and development, along with current and historic coal mining influence. We found a significant difference between nail and blood concentrations based on mean concentrations for each tissue type and a significant Kruskal-Wallis test, which was expected because of the different snapshots they provide. Blood being short-term accumulation, whereas nails are more long-term accumulation. Mercury levels are indicated in the figure as the points, with the lower tails indicating methylmercury levels, which is estimated to be within 70 to 100% of detected mercury in turtles. All samples exceeded the safe consumption limit set by the EPA, while methylmercury exceeded the fish criterion limit in 18 of the nail samples. Although nails are not consumable parts of the turtle, they are positively related to parts of the turtle that are typically consumed, such as muscle and liver tissue. Therefore, consumption advisories should be implemented to minimize possible health and safety concerns. With that, I would like to thank my committee, funding sources, interns, volunteers, and landowners for providing support in this study, and I will gladly take any questions you might have. Thank you so much.